Our next speaker will be Dr. Bafor from National Cancer Institute at the National Institute of Health. And the talk will be titled Overexpression of Interferon Gamma Suppression that suppresses double negative Tregs and functions in autoimmune ovarian failure. Dr. Bafor, with that, I would like to hand it over to you. Thank you very much for joining us this morning.
Thank you for the opportunity to be here. So I'll start by sharing my screen. Okay, hello everyone. I'm Toma Bafor of the National Cancer Institute. I will be presenting a part of my research, which I've titled Overexpression of Interferon Gamma Suppresses Double Negative T Regulatory T Cell Function in Autoimmune Reproductive Failure. And my mentor is Dr. Howard Young, no conflicts of interest declared. Now, ovarian autoimmunity is actually immune response targeted to the ovaries, such that they lose function or fail completely. And it's actually more prevalent than it's stipulated in some literatures, because as I have depicted here, we find that in women less than 40 years, you have ovarian autoimmunity, though a broad term because there are many other subtypes affecting over 1 million women. Now, if you put that into context, that reproduction itself is finite, meaning you have it occurring within a specific age period, from after puberty to, just before menopause. And so when you take that into context, you see that this is actually quite prevalent. And when we move that age further into the quarter of about 45 years, we find ovarian autoimmunity affecting about 5 million women in the United States alone. Now compare that with all other autoimmune diseases that occur in women in the United States, and that tells you that this is actually a relevant condition and one that requires a number of investigations into the cellular and molecular mechanisms that are mediating ovarian autoimmunity, even though, like I said, it's broad, because we can then begin to talk about all of the different subtypes involved. But this is necessary for us to, help improve the condition and also, develop new knowledge about the condition as well. And so to do that, in our laboratory, we developed a model. Our laboratory developed a mouse model, the AU-rich element knockout model. And they did this by deleting a conserved region in the interferon gamma gene, which is present both in humans and mouse. And it's within the 3' UTR. So the AU-rich elements in that region was deleted and replaced with random nucleotides. And this led to an overexpression of interferon gamma. So basically, there were high levels of interferon gamma levels in the ARI knockout mice. And this AU-rich element knockout mouse, which I will refer to as the ARI mice from now on, is accepted as a preclinical mouse model of chronic inflammation and of systemic lupus erythematosus. Now, in our laboratory, we systematically characterize autoimmune ovarian failure in these mice. I will not go into much in the details of that, but we'll be happy to take questions afterward if necessary. And we are using that to investigate further the conditions, cellular changes that are occurring, and look into how we can translate this further as well. And so we discovered that there was increased autoovarian antibodies in our mouse, of course, indicating that there was immune response to the ovaries. And corresponding to that, we found increased cytotoxic T cell infiltration in the ovaries as well. And the subsets of the CD8 T cells were dominantly effector memory and central memory and phenotype. And we know that this is relevant in progressing the pathology, because these effector memory cells will recruit more circulating cytotoxic T cells from the circulation into the ovaries and worsen the condition. And so to confirm that the CD8 T cells were in fact mediating this condition, what we did was to transfer CD8 T cells from our ARI knockout mice into RAG deficient mice. And what we found was they replicated the disease. So we had the same autoimmune ovarian failure occurring in the RAG mice, as long as all of the other different conditions associated with it, including hormone changes, as well as pro-inflammatory cytokines as well being increased in these mice. And then we also showed that that when we depleted the CD8 T cells in the heterozygous RE mice, we found that reproductive cycling improved significantly from the treated to from those that were depleted of CD8 T cells to those that were depleted of CD8 T cells. We found improved reproductive cycling as well as improved numbers of reproductive cycle. Now, this is relevant because reproductive cycle is a reflection of the function of the ovarian status. And so when there is improved reproductive cycling, it tells us that the ovaries is also being improved. Now, all of this show to us that the CD8 T cells are driving the overexpression of interferon gamma within the ovaries and, of course, worsening or mediating that condition. Now, in addition to the increased cytotoxic T cells that we found in the ovaries, it was also necessary, of course, under the context to examine if there was breakdown in peripheral tolerance. And so we did find that there was a decrease in the FOXP3 gene expression in the ovaries. And of course, what this meant was that, the CD4 Tregs weren't functioning as they should within the ovaries. But in addition, and very interestingly as well, we found that a group of cells, the double negative T cells, which we see here in high levels in the wild type ovary, was decreased in the RE knockout ovaries and significantly so in the RE knockout. So we find this very interesting, significantly decreased frequencies of double negative T cells. And you see that there was a significant increase in the ratio of cytotoxic T cells to DNT rates in the ovaries. Now, before I go on to talk about, what we looked at in terms of these double negative T cells, because this was interesting, where we kind of wondered what exactly is happening to these DNT cells. Why are they being reduced? Are they not functioning as they should? I'm just going to share a bit about double negative T cells. Now, I'm not talking about double negative T cells within the thymus. These are double negative T cells in the periphery. They are mature and they're able to participate in immune activity. And specifically as regards this presentation, I'm referring to the regulatory subtype of these double negative T cells that express the TCR alpha beta. Of course, they're positive for CD3. They do not express the CD4 and CD8 co-receptor molecule, and they're negative for NK markers as well. They do release or produce a very interesting set of cytokines, which are somewhat different from what we have with the CD4 Tregs, but as concerns these presentations, I'll just leave it to these major ones, which is the secretion of interferon gamma, TNF alpha, and IL-10. Now, also, the DNT cells mediate their function more through cell-to-cell contact and through a process known as phagocytosis, where they acquire antigens from antigen-presenting cells and present same to the effector cells, where they then induce activation-induced cell death through the fast mechanism and, of course, apoptosis. And so we were very interested in why these DNT cells, which appear to be in high numbers in the ovaries and would suggest that they are critical to the function of maintaining tolerance within the ovaries as well. What's happening, why they're reduced and possibly not able to function as well, considering the exacerbation of the disease that we were seeing in our mice in the reproductive system. And so we fast sorted double negative T cells from wild type mice and then activated them and then co-cultured them with CD8 T cells while exposing them to different concentrations of interferon gamma. And one of the things we found was that DNT cells in the presence of interferon gamma while co-cultured with CD8 T cells, and the division was reduced, which is also seen here in red. So proliferation was reduced in the presence of interferon gamma, but which of course suggests that the presence of interferon gamma reduces the proliferation of DNT cells, and which will of course decrease their numbers. And we also saw that in the presence of interferon gamma, DNT cells were killed faster. We compared that with what was happening at the same time with the CD8 T cells, and we found that while DNT cells were able to kill CD8 T cells, in the presence of interferon gamma, there was no further killing. Perhaps this is occurring because there is now fewer numbers of DNT cells present and are not able to potentiate further killing of CD8 T cells, or because interferon gamma was in itself mediating proliferation of the effector CD8 T cells. We're not quite sure, but we do see that the DNT's ability to kill CD8 T cells, effector T cells further, was hindered in the presence of interferon gamma. We were also interested in looking at the fast cell killing mechanism of DNTregs, what was happening with them in the presence of interferon gamma. Now, we looked at what was happening with CD8 T cells, interferon gamma, the different concentrations increased, fast cell expression in CD8 T cells, but with this DNT cells, we did see a decrease in fast cell expression, suggesting that these killing capacity of DNT cells was decreased in the presence of interferon gamma, which is shown here in purple and pink. And then we also looked at fast receptor as well. We know that interferon gamma generally will cause, fast receptor expression in cells. And while, yes, it increased CD95 expression in CD8 T cells, which is shown here, we also see that significantly increased fast receptor expression in the DNT cells. And what this implies is that there will be increased autologous killing of the DNT cells and also increased CD8 T cell mediated killing of DNT cells, which will all work together in reducing the numbers of the DNT cells present in the tissue as well. So considering that, okay, fine, we do see and that the major killing mechanism of the DNT cells have been kind of reduced in the presence of interferon gamma, we also wondered, what about, the suppressive cytokines? How are those affected? Interestingly, we found that in the presence of interferon gamma, there was further increase in interferon gamma production by these cells, but also we found a decrease in TNF alpha, and we know that with regulatory subtypes, TNF alpha will interact with the TNF-R2 receptors to induce suppression, and then we also found a decrease in IL-10 production as well. Now, we're not sure why there was further increase in DNTregs, and we're not quite sure how DNTregs are utilizing interferon gamma in mediating their regulatory function, but perhaps there is a sort of threshold where, with interferon gamma, where DNTregs can function, and over and beyond that is actually counterproductive to DNT cells. But we intend to look at this further. But what we see overall, as I like to summarize here, is that CD8 T cells are driving that interferon gamma expression in autoimmune ovarian failure. And with that actually increases fast receptor expression in the DNT cells, that also decreases the fast L expression in the DNT cells. And what all of this does to these regulatory cells, of these unique regulatory cells, is it will decrease their proliferation, it will increase their apoptosis, and also decrease the suppressive cytokines that they produce. And so in conclusion, what we see is that the chronic interferon gamma expression, which we see is driving autoimmune ovarian failure mediated by CD8 T cells, actually act to suppress peripheral tolerance mechanisms, in particular suppressing double negative T cell numbers and functions. And by modulating this new knowledge that we have, we can actually try to improve how DNT cells can improve their function under the context of autoimmune ovarian failure. And with that, I would like to acknowledge every member of my lab, particularly Dr. Howard Young, who is my PI, and as well as all of the members of the core that have facilitated the success of this research. Thank you very much.
That was a really wonderful talk. Thank you so much for that, Toman. Really appreciate your time and the very interesting topic, actually. My PhD was in gamma delta T cells, so when you said DN T cells, immediately I thought, oh, these must be gamma deltas, but turns out I learned about something completely new. I'm curious if you could comment on the presence of these cells in, other tissues, and also whether, for example, during a chronic infection, their numbers increase, as an example being, let's say, in the context of COVID.
Oh, that's interesting. So they are present in other tissues, as far as I'm aware. I know they're present in the kidneys, for instance. Of course, they're present in the secondary lymphoid organs. That's where they interact. I've not investigated to see much of the other tissues, but I would be surprised if they're not there as well. Now, you see, what we have is we have different subtypes of these double negative T cells in the periphery. And there are literatures that suggest that there are some of them that actually have cytolytic effects, which is and they seem to be, those ones seem to be sort of regulated by the FAST receptors as well, with the presence or the absence of either a FAST or FAST-L. And you find them more in the lymphoproliferative models. So they've been described in those models a lot, but these are not mainly the regulatory subtypes, perhaps. And it is assumed that those DNT cells in those models are actually driving that infection. But I think, the fact that they've been increased in a particular infection due to the mutations in the FAST receptors does not necessarily mean that they are actually driving the infection, because not just the DNT cells proliferated, all of the other T cells that depended on that also proliferated. So with regards to COVID, I really cannot say, because this is actually a very new cell that is, it's still under investigation. And one of the difficulties in studying this cell is because of the lack of markers, even though a very recent study, I think published in 2021, identified some transcription factors like IKZF and Ly6C, I think, that they believe may be responsible in mediating, differentiation of these cells. And I think until we do, we're still kind of, jogging on, trying to understand exactly the roles that these cells play in different conditions.
Thank you so much. A
Sure.
few more questions, if that's okay.
Sure.
One question from Mireille is if you can comment on the direct applications of, this topic in women's health. What could be some potential therapeutic implications?
Yes, so I actually love that question, because what we've not done it yet in the females, but we found that the males with this deletion actually also have some form of reproductive issues as well, and we'll be releasing a publication on that soon. But when we administered these double negative Tregs to the male mice, they actually improved and were able to become fertile. So we think that that same thing can happen with the females. We just haven't done it yet. And so I think what's happened is when you give the DNTregs, if you've been able to modify and activate them such that they are healthy and able to resist the inflammatory environment that they encounter, then they will be able to support, because that's one other thing they do. They're able to support the function of the other regulatory cells as well. And they are, and probably that's why they're in such low numbers. They're actually very small. And so they support that. And at the same time, they are able to stay longer than most other cells, and mediate, suppression of the other effector cells. So when we did transfer these to the males, and we found that males that were otherwise infertile, unable to, have litters, they began to have litters, and that's actually a very good success on the part. And we think that if we can translate this to females, that we can actually help improve, the ovarian failure, autoimmune ovarian failure in women as well. And just to quickly add, DNTregs is currently being used, I think, by Dr. Li Zhang, as an off-the-shelf therapy for leukemic patients. And we think we can therefore utilize this opportunity clinically and translate it for good use.
Great answer. I see Eleanor is on, so I believe she has a specific question as well. Go ahead.
Yes, so it was great talk to him, and indeed I was going to mention Dr. Zhang's work.
Yes.
And how it's clinically applicable. So do you have any, I know you said there aren't any markers that you can necessarily pick up on, but do you have any sense whether there are actually sex differences in the numbers of these in humans?
Sorry, could you repeat the last bit?
Do you have any sense where there is a sex differential in the numbers of these double negative Tregs?
That is a very interesting question. So I have not assessed the numbers in males, but I expect that there will be, primarily because I know that steroid hormones actually regulate the numbers and proliferation status of these cells. And I know that estrogen is quite dominant in doing that. And perhaps, because we have this dominant release of estrogen in females, perhaps there might be more DNT regs in females than in males, but I cannot speak to that definitely.
Because
that's interesting.
I'm just wondering whether that, is reflected, in the number of inflammatory disorders which are prevalent in different sexes.
Yeah, that's actually very likely. And we're seeing, and what's coming up is, in terms of this sex bias to autoimmune diseases, it's not necessarily estrogen, but testosterone that actually might be a differentiating factor here, which is something that we would also be releasing very soon.
Great talk. Thank you very much.
Yeah, thank you.
Thank you. We've asked you maybe too many questions, so I encourage you to please take another look at the Q&A section, as there are a few more.
Okay.
And thank you so much for joining us this morning. It's wonderful to hear about your work.
Thank you for the opportunity to be here.